Okay, so today's Bio 20 lesson is going to be on photosynthesis and specifically the two reactions that occur in photosynthesis. We have the light dependent and the light independent. I feel like we're going a little bit backwards because we already did cellular respiration in the last video. And if you're a plant, then you're going to want to do photosynthesis to create your glucose first. And then to break it down into usable energy, you would have to do cellular respiration after that. So that is okay as long as you can differentiate between the two. They are very similar, so I feel like the biggest struggle with learning these two units is the fact that they are so similar, and it is easy to get some things mixed up. All right, so we're going to figure out where photosynthesis occurs. So if you look at a plant cell, we have organelles in here called the chloroplasts, and then there's also a lot of cytoplasm suspending all the rest of the organelles inside the plant cell, wherever those may be. We have the chloroplast over here, and inside the chloroplast are these discs called thylakoids. So thylakoids are these discs that are formed in stacks, and the stacks of them are called granum. So we have granum, thylakoid. So these individual discs are called thylakoids. This is the chloroplast. And then inside the chloroplast, we have another liquid that suspends these stacks of granum, and this is called the stroma. So this liquid in here is called the stroma. And then inside the thylakoid itself, we have a liquid inside called the lumen. And really, that's all you have to know, except the fact that we are going to zoom in inside the thylakoid membrane. So this line here is the membrane of the thylakoid, and this is where we're going to zoom in for a lot of the processes. And that includes the next one that we are about to talk about. So here we are taking a zoom in on the thylakoid membrane. So if you look at the thylakoid membrane, it might look like this. And then over here we have the lumen, which remember is the liquid inside of the thylakoid. And then on the outside over here, we have the stroma, which is the liquid inside the chloroplast. Because if you can remember, the thylakoid is suspended in the chloroplast. So if this is a thylakoid right here, the stroma is on the outside over here. And this is the inside of the thylakoid called the lumen. Inside the thylakoid membrane, we have these complex proteins called photosystems. So there's two of them. There's one over here as well. I'm trying to save some space, but these are the two photosystems. And this one's actually called Photosystem II because it was actually discovered second, which is a little fun fact. But this is Photosystem I. So the two photosystems contain chlorophyll and other pigments that absorb light energy. So when sunlight strikes these photosystems, two electrons inside the photosystems then get excited. So they undergo electron excitation, and they start to move through an electron transport chain, which are these carrier molecules that carry electrons from one photosystem to another. And also has another role, which we'll speak about in a second. But this is what the electron transport chain might look like. So this whole thing is just one chain. And then there's also another one that's a little bit shorter that leads to the stroma. So this goes from photosystem 1 out into the stroma. So that's where the electrons are going. And then there's another molecule over here called ATP synthase, or another structure, I should say. So ATP synthase is an enzyme. And obviously, this is not what it looks like, but I'll try my best. It's like a little tunnel right here. And at the end, there's like what you could imagine as like a turbine. So a turbine kind of dipping into the stroma right here. Okay, let's just label this out real quick.
ATP synthase, which is an enzyme. And then we have the electron transport chain. So we say ETC. There's another electron transport chain, ETC. And the two photosystems as well. So this photosystem 2, I think this got covered by the arrow, and this photosystem 1.
Okay, so what's happening here is the sunlight, the light energy, is striking two electrons inside photosystem 2, and those two electrons undergo electron excitation. So then they start to move through this electron transport chain. And as it's moving through the electron transport chain, there are these hydrogen ions in the stroma that are now being actively pumped through. So there's these hydrogen ions over here, and it can't diffuse without this process. What it needs to do is it needs to get actively pumped through by the energy of the electron. So as it's passing through, energy is being released, and these hydrogen ions start to pump into the lumen. So the purpose of this is to build a high concentration of hydrogen ions in the lumen, and you'll figure out why in a second. Okay, so now we have a high concentration of hydrogen ions, or protons, inside the lumen, and the two electrons from photosystem 2 that were excited went through this electron transport chain, releasing energy along the way to pump hydrogen ions over here in the stroma to the lumen to build up that concentration. Okay, so now the two electrons ended up in photosystem 1, but they're not energized anymore. They released all that energy. So we want to focus on how do we get those 2 electrons back in Photosystem II before we continue with this process. So in order to make this happen, there's water inside the lumen that undergoes a process called photolysis or photolysis, whatever it's called. But the point is that the sunlight is now striking the water that's inside the lumen, and then this water splits into 2H, half an O2, and there should be a plus, and also 2 electrons. Okay, so what's happening here is that the 2 hydrogen ions are now also populating this lumen over here to keep building that concentration. And then this half an O2 leaves as a byproduct of this. So it's just like a happy accident, right? So it just leaves. So now this is leaving, and if you remember, the whole process of photosynthesis also has oxygen leaving as one of the products. So this is where that's coming from. And the 2 electrons are now moving into this photosystem. So then they could get re-energized or re-excited and then move through this ETC. And it's probably pretty obvious that the sunlight is going to strike it again in Photosystem I, and then they're going to move through the second electron transport chain. Now you're right, but we do need to focus on these hydrogen ions first. We'll get to that in a second, but we need to focus on why we built this concentration gradient. So there's a high concentration in the lumen, specifically because these hydrogen ions need to move through what's called ATP synthase. So these hydrogen ions are passively diffusing through ATP synthase. And as it's doing this, the ADP, so ADP is the unenergized form of ATP, and we probably used up the ATP, which is why we got the ADP, and the phosphates, okay? So these need to turn into ATP, and the way that it does this is because of the physical movement of those hydrogen ions moving through. So one hydrogen ion moving through will force this ADP and the phosphate to form ATP. So these basically spin around like a hydroelectric dam, and it creates ATP. So it's physically forcing these two together. It's not a part of the chemical equation, it's physically forcing it together, and at the end you also have hydrogen up here. Okay, so we have the ATP now, and we have the hydrogen ion up here. This hydrogen ion is now important for the next step, so we can focus back on the 2 electrons and what happened to them. Okay, so now they get re-excited by sunlight, just like you probably guessed. And then because of this, they start moving through the second electron transport chain. And this time we're not trying to pump in hydrogen ions, but what we're trying to do is we're trying to get the electrons inside the stroma. Okay, so inside the stroma, there's also NADP+. So NADP is the oxidized form of NADPH. So we probably used it in one of the other processes and it turned into NADP. Okay, so this is the same thing that we had with ADP and phosphate over here, right? So we used the ATP in another process, and the ADP and phosphate are now left over, and we're trying to get more energy and we're trying to get more electrons. Okay, so what's happening here is the 2 electrons are moving over here where the NADP plus is, and as well as the hydrogen ions that were pumped through the ATP synthase, and they weren't used in this chemical equation, so they're just left over. But now they move over here to bind with the NADP plus and the 2 electrons. So these 3 are now coming together to form a chemical equation. So you have NADP plus plus a hydrogen ion plus 2 electrons. Okay, and now that makes NADPH inside the stroma. Okay, so we made ATP in the stroma now, and we made NADPH in the stroma. So as a summary, to get our ATP as well as our NADPH as our final products, we had to have a proton pump, so these electrons moving through this electron transport chain, pumping the hydrogen ions from here to here, and then we had those hydrogen ions build up a high concentration, and then it moved through a semi-permeable membrane called ATP synthase, and then that created ATP by physically turning a turbine. Okay, so what that process is called chemiosmosis. So we had the proton pump and chemiosmosis, and as a result we had the electrons moving out through here, binding with NADP as well as hydrogen ions from this process over here, making NADPH, and then we had ATP because we had the chemiosmosis occurring. So these are our 2 final products inside the stroma, and they carry on to the Calvin-Benson cycle. So we're going to use these 2 products in order to make glucose. I just realized how messy this diagram was. I feel like it would have been a lot better if I just used the notes like last time, but you know what? It is what it is. Here in the stroma, we have the Calvin-Benson cycle. So from the last process, we got ATP and NADPH. Okay, so those were our 2 final products. I'm actually going to try to make this neat this time, but there are 3 different steps in the Calvin-Benson cycle.
One is carbon fixation. Okay, and the other one is called reduction. And then over here we have regeneration.
So already inside the cycle, we have a 5-carbon molecule called RuBP. So we're going to regenerate this after going through the whole cycle, so it's already in here, okay? So what we're doing over here is exactly what it's called. The first step is called carbon fixation, and we need at least 3 carbon dioxides in order to keep this cycle going. So that means we need to do the cycle 3 times in order for anything to keep occurring, okay? So we need 3 CO2s, and then these 3 CO2s have 3 carbons, right? So now we have these RuBPs, so there's going to be 3 of them since we're going through the cycle 3 times. We have these 3 RuBPs, now gaining an extra carbon. So 1, 2, 3, 4, 5, 6. So each of these RuBPs gain an extra carbon. So this times 3. This is future me from the video. I am cutting into the video because I forgot to mention there is Rubisco, which is an enzyme that catalyzes the CO2 and the RuBP, forming the 6-carbon molecule. So the Rubisco is needed in order to catalyze this reaction to happen. Okay, and now because there's 6 carbons, it is now easily split in half because it's so energized. So now we're left off with 6 of this 3-carbon molecule. And this 3-carbon molecule is called 3-PGA. So 3-PGA, we have 6 of them because we went through the cycle 3 times. Okay, so we have 6 of these molecules, and now we need to go through the second step called reduction. We are going to reduce this 3-PGA. Okay, but first we need to bring in some ATP. So we're bringing in times 6 ATP, 1 per 3-PGA. And since we have 6 of them, we need 6 ATP. So we donated 6 phosphates because we had 6 ATP. And then those ATP turn into ADP. Now those 6 ADP return to the light-dependent reactions in order to turn into ATP again. So that's why we have these 2 processes inside photosynthesis. So we have our 6 ATP, a donated phosphate, and now the molecule looks something like this. But in Biology 20, we kind of skip over this step, and we just look at the fact that it's also getting reduced at the same time. So let's get rid of this. So you have these 6 ATP coming in, and at the same time, you also have 6 NADPH coming in as well. So you have, I know I promised this wasn't going to be messy, but I'm going to have to break that. So this NADPH is actually the reason why we don't have that extra phosphate anymore. So now the molecule still looks the same as before. We don't have that extra phosphate at the end, right? Because the ATP donated that phosphate, but because of the NADPH, we don't have that phosphate anymore. In Biology 20, you don't have to worry about that. We just go over the reduction phase and then the result of the reduction phase, which is that. So this result is called G3P. So G3P is just very energized. It's like the energized form of 3-PGA. And we have 6 of them. So this NADPH was then oxidized and turned into NADP. And it has to return to the light-dependent reactions to then gain electrons and hydrogen ions. So now we are left with 6 of these G3Ps. So there's these energized forms of these 3-PGAs. So the G3P that we just made, one of them leaves in order to make glucose. And we need 2 of them to make glucose, but the reason why we're not using 2 of them out of the 6 that we made is because of the regeneration phase. So we're going to explain that in a second. So one G3P goes on to make glucose, but it can only make half a glucose, so it doesn't actually make anything. So there's a G3P, and it goes on to make glucose, but you need 2 of them, okay? So what happens to the rest of the 5 G3P is that they regenerate. So these G3P times 5 then go on to regenerate. But if you look at the G3P, this doesn't look nearly like RuBP yet, right? There's 3 carbons over here, and there's only 1 phosphate. However, if you rearrange these carbons, if you look at it like this, you have 3 carbons per molecule and you have 5 molecules, which equals 15 carbons total. But that also means you could have 5 carbons per molecule and 3 molecules, which also equals 15 carbons. So that is exactly what we do. We rearrange this molecule to look something like this. And by the way, I made this all look a little bit better for everyone's sake. But now we have 3 of these molecules and they have 5 carbon each, which still equals 15 carbons. So we didn't actually change anything, we just rearranged it. So we still have the same amount of everything, but it looks a lot more like this RuBP molecule. But we're still missing one thing, and we're missing that phosphate. So if you look over here, we have 2 phosphates, 1 on each end, but over here we only have 1 phosphate. So what we do is we add 3 ATP, and then we get 3 ADP from that, which need to go to the light-dependent reactions to get recharged. But basically, we're giving those 3 phosphates to these 3 molecules, and then we end up with RuBP again. So RuBP is the end of it, and then we restart the cycle all over again. However, this only made half a glucose, which really means it made nothing. So therefore, we need to multiply all of this by 2 in order to make one full glucose. So I highly recommend memorizing this, because if your teacher asks for one G3P molecule, which is half a glucose, this is what everything would look like. This is what the numbers would look like. But if your teacher asks for a glucose, all you have to do is just multiply it by 2. So this is what we're going to do. We're going to add a red marker, and we're just going to cover over all of the multiplication signs in order to count for one glucose. So instead of 3 RuBPs at the start, we're actually having 6 RuBPs, because we're going through this cycle 6 times. Not because there's 6 going through at once, but we're going through the cycle 6 times, which also means we need 6 CO2. And that also means we end up with 6 of these 6 carbon molecules, because we added 6 CO2 to the RuBP. So you end up with 6 of these, and then once you cut them in half, you end up with 12 of these 3PGAs, okay, because they're split in half. So now we end up with 12 of these, and then we bring in 12 ATP, because we need 1 ATP per 3PGA, and we also bring in 12 of these NADPH. So these both then turn into their oxidized, unenergized forms. So then they need to go to light-dependent reactions in order to re-energize and get reduced. So then we go to the G3Ps, and we now have 12 of these, because we energize and reduce the 3PGA, and now we have 12 of these G3Ps. And instead of having only 1 going towards making glucose, we can afford having 2 going towards making glucose. And 1 glucose only needs 2 of these G3Ps, therefore we make 1 full glucose molecule. So now 10 can move on and regenerate to this RuBP. So let's look at the numbers now. So you have 10 of these 3 carbon molecules, and if we rearrange it, we have 3 molecules per G3P times 10 of the molecules itself equals 30 carbons. So we have 30 carbons, which also means we could do 5 carbons times 6 molecules. So we end up with 6 of these molecules over here, and there's 5 carbons in each molecule, which still equals 30. Okay, so now this is times 6, because we always want those 5 carbons inside the molecule, which is why we multiplied it by 6. But we end up with 30 carbons at the end. So you have 6 of these molecules, and they're almost like RuBP, but they're missing the phosphates. So we now need to add 6 ATP. From that, we get 6 ADP, which now need to go to light-dependent reactions. And then we end off with another 6 RuBP. So this goes back to the start, and we start off with 6 again. So that is the whole process, and we made 1 glucose, but what we needed is we needed 12 ATP plus the 3 ATP from, or the 6 ATP from here, and then we needed 12 NADH, and we also needed 6 CO2. So 12 plus 6 equals 18 ATP total is what we needed. So that is how much we made inside the light-dependent reactions in order for this to occur. And then we also need 12 NADH from the light-dependent reactions for this to occur. And then we needed 6 CO2. So if you remember the photosynthesis equation, we had 6 CO2 plus 6 H2O equals C6H12O6, which is a glucose molecule, plus 6O2. So the 6O2 came from the light-dependent reactions, because remember, we were splitting water and O2 came as a byproduct. And then we have 6 CO2 from this over here, because we ended up using 6 CO2s for 6 cycles. And then the glucose came from this whole process right here. We needed to make 2 G3Ps for it. And that explains just about everything in the photosynthesis equation. So now we know how exactly all of that happens. So to end this off, we have this entire summary of what just happened. So we have the light-dependent reactions. We needed light to split water in order to make O2, and also we needed to make ATP and NADPH as our end products. And then those end products went into the Calvin cycle inside the stroma, where CO2 came in, and then it turned into G3P, which turned into glucose, and then that could either turn into starch or sucrose. So this is all happening inside the chloroplast. The Calvin cycle is happening in the stroma, but then we also have some dipping into the lumen. So I would like to thank you guys for joining me again. I really hope you guys do good on your quizzes. I will post more videos just like this in the future, just so I can have a better and improved version of these videos that I'm making. But I have my unit test coming up, So hopefully we all do good, and I hope this video helped you. So leave a like and definitely subscribe if you want more future content.